TIM4 affinity methods targeting phosphatidylserine for isolation or detection of extracellular vesicles, PS affinity methods. Rikinari Hanayama, WPI, Nano Life Science Institute, Kanazawa University First. Let me briefly explain my research background before coming to EV research. I used to work on the molecular mechanisms of phagocytosis of apoptotic cells. When cells undergo apoptosis, the apoptotic cells present an eat me signal to macrophages, and macrophages recognize the signal using specific receptors. The best known eat me signal is an anionic phospholipid of the cell plasma membrane, phosphatidylserine. We identified MFGE8, also known as lactoferrin, and TIM4 as potent phosphatidylserine receptors that promote phagocytosis of apoptotic cells. When we established MFGE8 knockout mice, we found an interesting phenotype. MFGE8 was originally identified as a protein expressed on the surface of milk fat globules, a kind of extracellular vesicles secreted in milk. Upon weaning, the developed mammary glands undergo involution. The MFGE8-KO caused the accumulation of a large amount of milk fat globules in the mammary ducts, indicating that the excess milk fat globules are cleared by MFGE8. As the milk fat globules are large enough, we performed flow cytometric analysis of single vesicles. Milk fat globules from the wild-type mice expressed MFGE8 and also bound by annexin 5, which binds to phosphatidylserine. In contrast, milk fat globules from MFGE8 knockout mice were of course not expressing MFGE8, but strongly bound by annexin 5. These results indicate that milk fat globules expose phosphatidylserine on their surface, which are bound by MFGE8. In other words, relatively low binding of annexin 5 to wild-type milk fat globules is due to the masking of phosphatidylserine by MFGE8. In another work, we identified TIM4 as a potent phosphatidylserine receptor that promotes phagocytosis of apoptotic cells. However, when we established the BAF3 pro-B cell line expressing TIM4, we found the annexin 5 strongly bound to the cells, but not to the parental cells. We thought this is weird and performed EM analysis. We found that the TIM4 expressing cells were associated with many EVs, which exposed phosphatidylserine on their surface, as revealed by the binding of annexin 5 gold particles. Then, what is the mechanism for phosphatidylserine exposure in EVs? We still don't know the exact mechanism yet, but here's our hypothesis. It is known that phosphatidylserine is exclusively kept in the inner leaflets of lipid bilayers of healthy cells by the action of type 4 p-type ATPase. When EVs are separated from the cells, the ATP is no longer provided from the mitochondrial energy-generating system. Therefore, the exhaustion of ATP might be the cause of PS exposure by inactivating the ATP-dependent flippase. Based on these results, we thought that TIM4 might be useful for a high-purity isolation method of EVs. As the binding of TIM4 to the phosphatidylserine on the surface of EVs is calcium-dependent, intact EVs can be easily released from TIM4 conjugated magnetic beads by adding calcium chelators such as EDTA. This novel affinity-based EV isolation method has been available as a research kit from Fujifilm Wacko, whose name is a MagCapture Exosome Isolation Kit PS. We believe this is one of the best methods to isolate highly purified EVs. And now I will show you the supporting data. Here is a simple experiment. Human cells were cultured in the medium containing 5% fetal bovine serum in which EVs had been depleted by ultracentrifugation and PEG precipitation in advance. The conditioned medium containing both EVs from human and proteins from bovine were subjected to the standard differential centrifugation to obtain 10K supernatants, which were also filtrated. Then, small EVs were isolated from the 10K supernatants either by our TIM4 method or ultracentrifugation. The TEM analysis revealed pure SEVs isolated by the TIM4 method. On the other hand, a large number of small precipitates could be observed by the ultracentrifugation. In addition, the mean size of SEVs isolated by the ultracentrifugation was larger than that isolated by the TIM4 method, probably due to the increased populations of aggregated or fused SEVs larger than 200 nanometers. And Western blot analysis revealed about 10 times more enrichment of SEV markers such as CD63, CD9, and CD81 by the TIM4 method. We confirmed that this is also true to the SEVs from various cell types. Residual SEVs after TIM4 method were hardly isolated by ultracentrifugation, indicating that TIM4 method had isolated most of SEVs. The enrichment of SEVs was further confirmed by density gradient fractionation. Western blot against CD63 revealed the enrichment of SEVs in the third fraction, which corresponds to a density of 1.1 grams per milliliter. Most proteins isolated by the TIM4 method were enriched in this third fraction. On the other hand, many proteins were also observed in the fourth and fifth fractions by the ultracentrifugation, which are not from SEVs. Here's a list of top 15 proteins identified in the isolated SEVs by shotgun proteomics analysis. White columns are human proteins mostly from SEVs, and gray columns are bovine proteins from FBS. As you can see, many bovine proteins were identified by ultracentrifugation, whereas only one was identified by the TIM4 method, which bound to the magnetic beads. Most notably, many known SEV markers, such as several annexins, flotillins, and syntenins, were the top hits by TIM4 method, whereas many nuclear proteins such as DNA PK, helicase, and ribonucleoprotein were the top hits by ultracentrifugation, indicating that TIM4 method enriches much more SEVs. We also compared the enrichment of SEV microRNAs, qPCR analyses of MIR16, MIR92A, MIR1423P, in the isolated SEV fractions, revealed 3 to 4 less CT values by the TIM4 method, indicating that about 10 times more SEV microRNAs were isolated by TIM4 method. Here is a comparison of functional activity of SEVs. We examined how MSC-derived SEVs can suppress the gene expressions of inflammatory cytokines in LPS-stimulated monocytes. With the same particle number, the induction of TNF-alpha and IL-6 were more strongly suppressed by SEVs isolated by TIM4 method than those by ultracentrifugation. Next is a comparison of anti-fibrotic activity of SEVs. We examined how MSC-derived SEVs can suppress the gene expression of fibrosis markers in TGF-beta stimulated fibroblast cell line. Again, with the same particle number, the induction of collagen 3, collagen 5, and alpha SMA were more strongly suppressed by SEVs isolated by TIM4 method than those by ultracentrifugation. Now I hope you understand the advantages of using TIM4. However, I have been often asked the same question. What percentages of SEVs actually expose PS and can be isolated by TIM4? Of course, as is the case with other isolation methods, it is not 100%. To directly address this question, Dr. Matsumoto at Kyoto University has established luciferase expressing SEVs by fusing it with LAMP2. The bulk SEVs containing both PS-positive and PS-negative SEVs were subjected to the isolation by TIM4 method. The mass balance determined by percentage of luciferase recovered rates indicates almost 90% of SEVs were captured by TIM4 beads, and this is true to all cells they have examined so far. As I explained in the beginning of this talk, PS is a signal for phagocytic clearance. Accordingly, the PS-negative SEVs were much more resistant to clearance in serum after IV injection than PS-positive SEVs, especially not targeted to liver, where TIM4 is highly expressed by macrophages. In other words, PS-negative SEVs prepared by TIM4 method could be a powerful tool for EV-based drug delivery system. Another question is which subpopulations could be enriched by each isolation method. As you know, phosphatidylserine is an anionic phospholipid. We therefore subjected bulk SEVs isolated by ultracentrifugation to anion exchange column to fractionate SEVs based on differences in their anionic charge. When SEVs were eluted by increasing sodium chloride concentration, we could obtain three groups, which corresponds to neutral, low anionic, and high anionic SEVs, and each percentage is as shown. Anti-tetraspanin antibodies binding to each fraction revealed the first fractions contained few typical SEVs. On the other hand, the low anionic SEVs showed the highest tetraspanin expressions. Here is a comparison of the number of SEV subpopulations isolated by the three methods: ultracentrifugation, TIM4 method, and size exclusion using QEV. The TIM4 method yielded more low anionic SEVs, but less neutral and high anionic SEVs than the other two methods. Some researchers claim that exosomes are more enriched in the low anionic fractions, whereas microvesicles are on the high anionic fractions. This might be the reason why MSC-derived SEVs isolated by TIM4 method had stronger functional activities. Okay, please, let me move on to the second topic. One of the major advantages of TIM4 method is the ability to perform consistent data analysis in conjunction with the sensitive quantification of EVs by ELISA. Because of the strong ability of TIM4 to capture EVs, we considered the possibility that it might be applied to sensitive quantification of EVs by sandwich ELISA. Here, proteins known to capture SEVs, such as TIM4 or anti-CD63 antibody, were coated on a microtiter plate, and various concentrations of SEVs were allowed to be captured by them. The captured SEVs were detected by biotinylated anti-CD63 antibody followed by HRP conjugated streptavidin. As expected, TIM4 captured SEVs much more efficiently than other capturing antibodies, enabling about 1,000 times more sensitive and linear dose-dependent quantification of SEVs by ELISA. This has been available as PS Capture Exosome ELISA kit from Fujifilm Wacko. We confirmed that this sensitive quantification of SEVs can be applied to various cell types. Again, TIM4 can capture SEVs much more efficiently than other capturing antibodies. Although SEVs are heterogeneous, TIM4 can capture most subtypes of SEVs regardless of their surface markers by binding to membrane. Thus, we found that the expression patterns of tetraspanins are quite different among tumor cells, indicating that TIM4 ELISA is more sensitive and unbiased method to analyze various SEV subpopulations than using anti-tetraspanin capturing antibodies. By taking advantages of this method, we perform genetic screening for regulators of SCV secretion by introducing shRNA library into K562 leukemia cells or gene trap vector into HAP1 haploid cells. After examining changes in the amount of SCVs by TIM4 ELISA, we identified several candidates, including TSG-101. As you can see, TSG-101 knockout significantly reduced the amount of SCVs, whereas non-secreted vesicles accumulated inside the cells. Microscopic observation confirmed the accumulation of vesicles, shown in green color, in the TSG-101 knockout cells, which is surprising for us because TSG-101 has been implicated in the generation of exosomes as a subunit of ESCRT1 complex. By using a super-resolution microscope, we confirm the accumulation of non-secreted vesicles, this time in red color, in the TSG-101 knockout cells, especially at the edge of the cells, implicating that there might be hotspots for exosome secretion. From these results, we concluded that TSG-101 is a critical regulator of exosome secretion. Therefore, we tried to identify its partner proteins by immunoprecipitation and mass spectrometry. Based on the molecules we identified, this is our current working model. We found TSG-101 is not involved in the generation of vesicles inside MVE. Instead, TSG-101 might mediate the transport of MVE and its docking with the plasma membrane by interacting with partner proteins, as illustrated here. Thus, TSG-101 knockout impairs these processes, causing the accumulation of non-secreted vesicles. The secreted exosomes are captured by TIM4 or MFG-E8 by binding to phosphatidylserine and then uptaken by the recipient cells. Now, we can manipulate exosome secretion and uptake by targeting the key regulators such as TSG-101 and TIM4. Here is one example. As tumor-derived exosomes have been implicated in tumor metastasis, we established osteosarcoma cells deficient in TSG-101. As you can see, TSG-101 knockout did not affect the growth of the primary tumor cells both in vivo and in vitro. However, the occurrence of lung metastasis was dramatically reduced by TSG-101 knockout, proving that tumor-derived exosomes actually promote tumor metastasis by a loss-of-function experiment. Like this, we have recently found several mechanisms that promote tumor progression by tumor-derived SCVs. Specifically, we found that SCVs from osteosarcoma suppressed osteoclastogenesis by transferring MIR146A5P, which enhanced lung metastasis. We also found that SCVs from glioma-enhanced angiogenesis through down-regulating thrombospastin-1, a negative regulator of angiogenesis in microglia by transferring WT1. Today, I introduce the advantages of using TIM4 for high purity and intact isolation of EVs, and also for high sensitive and direct detection of EVs with easy operation and high reproducibility. In the last few minutes, I would like to add that TIM4 is also useful for flow cytometric analysis of EVs. As I explained, the TIM4 beads can capture most subtypes of SEVs, regardless of their surface markers, by binding to the membrane. After staining with antibodies, it is important to remove unbound antibodies by washing. TIM4 beads allow easy on-site washing without loss of EVs. Then, not only whole particles, but also single particles can be analyzed by releasing from TIM4 beads with EDTA. We used Nano FCM for the single particle analysis of SEVs. When WT SEVs prepared by TIM4 beads were double stained for CD9 and CD63, 4 distinct subpopulations were clearly separated. On the other hand, WT SEV prepared by ultracentrifugation showed a decrease in the double positive subpopulation and an increase in the double negative subpopulation. With CD63KO SEVs, TIM4 beads nearly eliminated the appearance of CD63 positive SEVs, whereas ultracentrifugation left 18%, resulting in a strange staining pattern. Similarly, with CD9KO SEVs, both methods nearly eliminated the appearance of CD9 positive SEVs, whereas intact CD63 positive SEVs were only detectable by the TIM4 beads, not by ultracentrifugation. Taken together, these results indicate that TIM4 method is more suitable for SEV sample preparation for FCM analyses. Finally, I would like to acknowledge my colleagues, especially Takeshi Yoshida, for producing these data. Thank you for your attention, and I'd be happy to take any question and comment.